But if there is oxygen present, we're talking about eukaryotes here specifically because nothing else would have a mitochondrion, then the pyruvate enters the mitochondria. Immediately upon entering the mitochondria, pyruvate gets oxidized. It gets oxidized, broken down a little bit. A CO2 kind of gets peeled off of there, one carbon pulled away, and the remaining 2 carbons on that pyruvate get picked up by coenzyme A, and we get a new chemical product called acetyl-CoA. Acetyl-CoA can then enter the next step of aerobic cellular respiration, which is the citric acid cycle or the Krebs cycle. In both cases, we start with glycolysis. Glycolysis is an anaerobic process, and it has to be completed before we can move forward with the aerobic respiration processes. Okay, so if oxygen is present, then we move from glycolysis over to what's called the transition reaction, or pyruvate oxidation is how I usually like to say it. The transition reaction occurs the moment pyruvate enters the mitochondrion. It's going to be oxidized into acetaldehyde, and then that acetaldehyde is going to be picked up by coenzyme A, and it gives you a new chemical product called acetyl-CoA. Our coenzymes are organic carrier molecules that kind of ship things around the cell. They bring parts around. ATP is a coenzyme. It brings phosphate groups over to proteins that need some energy to do their work. NADH is a coenzyme. It brings high energy electrons on its little hydrogen here and acts as a reducing agent for chemical reactions. Acetyl-CoA is bringing this acetyl group in to what we call the citric acid cycle or the Krebs cycle in order to continue oxidizing it down and getting more and more and more energy out of it. So now we've just created this new object, right? We went from glucose to pyruvate to acetyl-CoA now. So let's say that there is oxygen present inside of the cell. So we're going to do aerobic respiration. First, we started with glucose. Glucose is a 6-carbon molecule. Through glycolysis, we broke it into 2 pyruvates. Okay, 3 carbons each. The process there was glycolysis. Next, pyruvate enters the mitochondria. So we finally exited the cytosol and we're now in the mitochondria, and immediately it gets oxidized by enzymes inside the mitochondrion. Now, when I say oxidized, in this case, I'm not just talking about losing electrons. I'm going to actually remove a carbon from it, and I'm going to slap oxygen onto that carbon. So CO2 is produced. Right? And this happens a couple of times in the next few steps. We're going to oxidize it to release some energy here, and CO2 is going to be produced. We essentially pluck off 1 carbon and allow it to leave the system because we've gotten every ounce of energy we can out of the electrons in those particular bonds here. Now this 2-carbon molecule is acetaldehyde, but it's going to be picked up by something called coenzyme A. CoA. You guys know coenzymes. Coenzymes are those organic carrier molecules that kind of move materials around the cell for any kind of chemical reaction or metabolic process. So for example, we've seen other coenzymes before. ATP is a coenzyme. NADH is a coenzyme. They carry phosphate groups and hydrogen atoms respectively here. So coenzyme A, what's its job? Well, its job is to carry these carbons into the Krebs cycle. The Krebs cycle, or the citric acid cycle. It's named after one of the chemical products of that particular pathway. So the Krebs cycle is what we're going to talk about next. Now, in the Krebs cycle, acetyl-CoA is going to be oxidized even further. We're going to make more CO2, right? And this makes sense because this is cellular respiration. It's all about taking in oxygen and breathing out CO2, at least at the macroscopic level. But on the cellular level, we have to actually use that oxygen to break down this organic molecule, and slowly but surely, each carbon is going to be stashed away into CO2 and removed from the system. It's going to exit there. Now, in the Krebs cycle, we're not going to make a lot of ATP. We'll make a couple. We'll make a couple ATPs. But what we're mostly going to be doing is making other kinds of coenzymes. We're going to make more NADH, a lot more NADH. We're also going to make another one called FADH2. Be patient with this. Eventually, we're going to convert all the energy in these coenzymes over to ATP, but that's the very last step. If you look down at that slide, you can see item number 3 is going to be another electron transport chain. This one's called oxidative phosphorylation, and that's where we're actually going to take all the energy from those coenzymes and eventually get to ATP. So ATP is going to come eventually. We're going to get lots and lots of ATP right at the end, but the point of the Krebs cycle is mostly to make these kind of intermediate coenzymes to stash the energy temporarily. Now, this slide here shows the important things in the citric acid cycle that you are going to want to know. You don't need to know every single chemical intermediate. Just like the Calvin cycle in photosynthesis, this is a metabolic cycle. So the products of one chemical reaction, of one enzyme's process, is going to end up being the substrate for the next enzyme's process in the chain. And this whole cycle creates a loop. It circles back on itself. Hence why we call it the Krebs cycle or the citric acid cycle. First, acetyl-CoA is going to enter this cycle, and then it's got to get locked on to another type of product. Again, metabolism is sometimes like solving a Rubik's Cube. You got to change groups and put them in the right shape and the right orientation with each other to get any kind of ultimate work done. Sometimes it takes a few different turns before you can actually see what the ultimate picture's going to be. So, acetyl-CoA comes in and gets locked onto a product called oxaloacetic acid. That is then chemically changed into citric acid, and the citric acid is then changed again and again. Now, there are 2 carbons left from the original glucose on each of these acetyl-CoA, which we have 2 because there were 2 pyruvates, so now we have 2 acetyl-CoA. Those 2 carbons are going to be stripped away during this chemical cycle, during this metabolic pathway. So those carbons are going to be lost as CO2. Okay. In the process of breaking it down, though, we're going to reduce more NAD. We're going to get more NADH. We're going to get 3 NAD plus reduced to 3 NADH. Right. Now, down here, we're also going to see some production of ATP. Finally, we're getting some more ATP in here. So we have ADP being converted to ATP. Now, you guys know that making ATP is called phosphorylation. When you add a phosphate group onto an ADP molecule, you've phosphorylated that ADP. And we had photophosphorylation, and we had substrate-level phosphorylation. Substrate-level phosphorylation is what we had in glycolysis here. This is more substrate-level phosphorylation. Over here, we have FAD being reduced to FADH2 in the process, right? And then eventually it comes back and we take new acetyl-CoA in and the whole cycle begins over again. But what was the point? What did we actually accomplish in total? Well, we got 3 NADH, we got an ATP, and we got an FADH2 for each acetyl-CoA molecule, of which there were 2. So the Krebs cycle, if we're taking this tally based on per glucose, a per glucose molecule basis, the Krebs cycle therefore gives us 6 NADH, 2 FADH2, and another 2 ATP. Whew. So what are we at here? We got, in total, 6 NADH created by the Krebs cycle, 2 NADH created by pyruvate oxidation, and 2 NADH created by glycolysis. In total, we have 10 NADH molecules and 2 FADH2s. Now, this next slide here walks you through the citric acid cycle step by step by step. I'm not going to demand that you've memorized every single step of the citric acid cycle, but some of you are going to go on to higher level biologies where they might actually want you to memorize each step and maybe even the enzymes that catalyze each step. If you find yourself in that position, there is a very silly story that I was told when I was a student, which I thought was just ridiculous when I first heard it, and yet I still have it memorized, and it's how I know the steps of the Krebs cycle, so I'm going to share it with you. So, there's a mitochondrion standing out on the street, and what does a mitochondrion do? It makes ATP, the energy currency of the cell, money inside the cell's economic framework, right? So it's making ATP, it's making money. And where's that money coming from? It's coming from glucose, right? So it's taking sugar and it's making money. It's selling sugar for money. Well, a police officer sees this. Here's my police officer, Sheriff Spider-Man comes in, and he says, "Hey, what is all this? I see you selling sugar for money there. Do you have the permit to be doing this? What is all this?" And he checks the mitochondrion to make sure that all of the mitochondrion's paperwork is in order and he's not cooking the books or anything like that. Everything works out okay, obeys the laws of thermodynamics, so the police officer is just going to write him a warning and knock off for the rest of the day. But then, the mitochondrion, he's still not sure about this situation, so at the end of the conversation, he says, "Officer, can I keep selling sugar for money?" Now, the first step of the citric acid cycle is that acetyl-CoA is going to lock on to a molecule called oxaloacetic acid here. That's the first step, okay? You should probably know that first step, right? Acetyl-CoA is going to combine with oxaloacetic acid, and they are going to produce the first byproduct of the citric acid cycle, which is, logically enough, citric acid, C. Officer Ken. That citric acid is then going to be chemically changed by another enzyme, which is going to give us isocitrate here. That isocitrate is going to be worked on by another enzyme, which is going to get us alpha-ketoglutarate. The K stands for the keto in alpha-ketoglutarate. This is also where we start making NAD for the first time. So we're going to reduce some NAD plus into NADH, which means that this product in the cycle is being oxidized. If NAD is being reduced, whatever it's working with has to be oxidized. Whenever you have an oxidation, you also have a reduction. And whenever you have a reduction, you also have an oxidation. So NAD is being reduced, and this isocitrate is being oxidized, and you get alpha-ketoglutarate. So, officer, can I keep? Next step is going to be to make succinyl-CoA. So coenzyme A actually leaves at the beginning of this cycle, but then it comes back and picks up this molecule in order to ship that to the next part of this equation. And at that point, we make yet another NADH molecule. Succinyl-CoA is then altered into succinate. And this is where we get some substrate level phosphorylation, creating an ADP. If you actually take a look carefully at that slide, it says that we make a GTP. Which is chemically very, very similar to ATP. It's just a different nitrogenous base on that coenzyme, but overall it's a very similar molecule, and that GTP can then be converted over to an ATP. So we'll just count it, we're going to count it as an ATP right there in order to keep things simple. So, officer, can I keep selling sugar? Now we're going to convert that succinate over to fumarate, another kind of acid, and in that process is when we create FADH2. So this is still being oxidized. The chemical product being passed around this cycle is still being oxidized, and now we've reduced FADH2. Every now and then I'm going to try to bring this back in order to try to make sure that you're following with it, because it's really easy to get lost in the language, but NADH and FADH2 are both energy-carrying coenzymes, as is ATP. So that's all we're doing. We're making different kinds of energy-carrying coenzymes. You do need to remember what kinds of coenzymes we make at each step in the cycle, but all of them have a very similar job. The only big difference is that ATP carries it in the phosphate group to phosphate group bonds, whereas FADH2 and NADH, they carry it on their high energy electron, which is on these H's, these hydrogens, right here at the end. Okay? So, officer, can I keep selling sugar for, and then malate is the next chemical product that comes in, and then malate is then converted over to oxaloacetic acid, and we get our third NADH up at that point there. You can see it on that chart. Whew! So that is the Krebs cycle in a nutshell. But let's look at the big picture here. The Krebs cycle, we have acetyl-CoA enter, carbon dioxide leaves. So now all of the carbon that was on that initial glucose that we brought in, it's gone. It's out of the system. We've gotten every ounce of energy out of that glucose that we possibly could, and we locked that energy into our various coenzymes at these other three processes. But the whole point of cellular respiration is to make ATP. So when are we going to make ATP? That's what's coming up right now.